There is one genetic fact about the origin of the Serbs that makes the standard story incomplete. Not wrong. Incomplete. Because what is usually presented as a single origin actually splits into two different genetic lines that do not fully agree with each other. For decades, the story sounded simple. A Slavic migration. A clear arrival. A people formed in one historical movement. That version feels clean. Logical. Easy to repeat. But DNA does not tell stories in straight lines. It preserves traces, contradictions, overlaps. And when scientists began to compare different types of genetic evidence, the simple origin started to crack, not loudly, not dramatically, but decisively. This video is not about myths or national narratives. It is about reconstructing a picture using data that does not care about tradition. We will take the genetic record apart into its main components: the paternal line, the maternal line, and the layers created by long-term contact with neighboring populations. Only then can we see what actually formed the Serbian genetic profile. And here is the core tension that drives everything that follows. One major genetic line points strongly toward a specific region and historical movement. Another does not follow that path at all. They intersect in the same population, but they do not come from the same place. That split forces a different question. Not where did the Serbs come from, but which part of the story are we talking about when we say origin? To answer that, we need to be precise about the tools. Because genetics is not one method. It is several independent systems that record history in different ways. The first is the Y chromosome. It moves almost unchanged from father to son. It ignores marriages, languages, and borders. Over long periods of time, it acts like a directional arrow, narrow, linear, and surprisingly stable. The second is mitochondrial DNA. This line passes from mother to child. It spreads differently. It accumulates variation differently. And it often reflects patterns of settlement and mixing that the Y chromosome completely misses. The third tool is ancient DNA, genetic material taken directly from archaeological remains. Not models, not reconstructions. Actual genomes from people who lived thousands of years ago in the same regions. Each of these tools is limited on its own, but when they are compared side by side, they reveal something uncomfortable. The Serbian genetic story cannot be reduced to a single migration or a single source. Before going any further, one rule matters. This is not about ranking populations, assigning purity, or rewriting identity. There are no better or worse lineages here. Only patterns and explanations that either fit the data or don't. Now, here's where the tension sharpens. When geneticists first focused on the paternal line of South Slavic populations, including Serbs, the signal looked surprisingly clear. Strong clustering, recognizable markers, a lineage that aligns well with known historical movements from the north into the Balkans. If that were the whole story, the investigation would almost end there. The Y chromosome would provide a clean anchor, a clear directional history, a satisfying conclusion. But genetics rarely allows clean endings. When scientists widened the lens and examined maternal lineages, the picture changed. Not slightly. Fundamentally. Different geographic signals appeared. Older layers became visible. Continuities emerged that did not originate from the same source as the paternal line. This is not a contradiction. It is a historical footprint. It suggests a process where incoming male lineages integrated into populations that already carried deep local ancestry through maternal lines. Not a replacement, not a reset, a merge. That realization alone reframes how origin should be understood. Because if one line points to migration and another points to continuity, then the population itself is the product of both processes happening at once. And this is where most simplified narratives fail. They choose one line and silence the other. There's another layer still missing, the regional context. The Balkans were never an empty stage waiting for a single group to arrive. They were a crossroads, a compression zone where populations moved, stopped, mixed, and adapted over thousands of years. Any group forming here would inherit complexity by default. Genetics confirms that expectation rather than erasing it. What looks shocking at first is not the presence of multiple ancestries. It is how clearly the different genetic systems preserve separate chapters of the same story. And here is the final loop that holds this entire video together. If the paternal line gives a simple answer, and the maternal line complicates it, then the real question becomes, which version do we instinctively accept as the origin? And why? Because the answer we prefer often says more about modern identity than ancient history. We will start with the paternal line, because it really does look simple at first. It gives a strong anchor, a sense of clarity, but that clarity does not last. If we strip the story down to its most direct genetic signal, we start here. Not with legends, not with names, with the paternal line, the Y chromosome. This is the cleanest tool genetics gives us. It passes almost unchanged from father to son, generation after generation. No recombination, no blending, over long time spans. It behaves like a historical arrow, pointing back through male ancestry with unusual clarity. That is why geneticists often begin here. And that is why, at first glance, the Serbian paternal profile looks almost reassuringly simple. When researchers compared Y chromosome markers across South Slavic populations, a clear pattern emerged. Clusters formed. They were not random. They aligned geographically and statistically with other Slavic-speaking populations of Eastern and Central Europe. This matters because clustering is not interpretation, it is structure. When thousands of samples fall into the same genetic neighborhoods, something real is being preserved. One lineage, in particular, appears again and again. It shows strong internal similarity. It expands rapidly in a relatively short time window, and its highest frequencies align with known historical movements into the Balkans during the early medieval period. This is where the simple story comes from. A male-driven migration, groups moving southward, lineages expanding, replacing or absorbing what came before. From the perspective of the Y chromosome alone, that story works remarkably well. Genetically, this lineage behaves exactly as a demographic expansion should. Low internal diversity suggests a relatively recent spread. Wide geographic reach suggests movement rather than long-term isolation. And its presence across multiple South Slavic populations points to a shared paternal origin rather than independent local development. If this were the only data we had, the investigation would almost end here. The paternal line would function as a genetic anchor. It would tie modern Serbs firmly to a broader Slavic expansion. It would provide a clean, defensible origin narrative based on measurable data. And this is why the Y chromosome became so influential in early discussions of population history. It delivers clarity. It reduces noise. It feels decisive. But clarity can be deceptive, because what the Y chromosome records is not a people. It records a specific pathway of inheritance, male to male, and nothing else. It does not track women. It does not reflect population size accurately. It does not record what happens when incoming males integrate into existing communities rather than replacing them. In other words, it tells us who arrived, not who remained. This distinction is easy to miss because the paternal signal is strong. Strong signals tend to dominate interpretation. They feel like answers. But good investigations test whether an answer survives when other evidence is added. That is where context becomes critical. The Balkans were not an empty landscape at the moment Slavic groups arrived. Archaeology, history, and earlier genetic layers all agree on this point. There were established populations, long-settled communities, deep regional continuity going back thousands of years. The Y chromosome does not erase that reality. It simply does not record it. So when we say the paternal line anchors the story, we mean something precise. It anchors one dimension of it. It provides a directional signal that aligns with migration, but it does not describe the full demographic process. This becomes clearer when researchers compare paternal data with regional diversity. The same Y lineage can appear across wide areas, while local genetic variation remains high. That combination suggests integration rather than total replacement. In practical terms, it looks like this. Male groups move into a region. They establish social dominance or demographic presence. Their Y chromosomes spread efficiently, but they do so within populations that already exist. From the outside, the paternal signal looks uniform. From the inside, the population remains complex. This is why geneticists are careful with language. They talk about male-mediated migration, not population erasure. They describe expansion rather than origin in the absolute sense. And this is also why the paternal line feels satisfying but incomplete. It answers a narrow question very well. Where do most modern Serbian Y chromosomes point? The answer is consistent and repeatable. They point towards Slavic-associated expansions, connected to Eastern and Central Europe. But that answer quietly avoids a larger one. Who was already there when those lineages arrived? To test that, we need a different genetic memory. One that spreads differently. One that is less sensitive to social hierarchy and more sensitive to long-term continuity. That is where the maternal line comes in. And this is the turning point. If we stayed with a Y chromosome alone, the story would feel almost finished. The anchor would hold. The narrative would stay clean. But the moment mitochondrial DNA enters the analysis, the simplicity starts to dissolve, because the maternal signal does not point as neatly in the same direction. And once that divergence appears, the idea of a single genetic origin stops making sense. That is where the investigation goes next. Now the picture shifts. When scientists turn from the paternal line to mitochondrial DNA, the geographic signal changes, and it changes immediately. The clean arrow disappears. In its place appears a layered pattern that does not point to a single arrival or a single source. This is the first real collision in the investigation. The paternal line suggests movement from the north. The maternal line often does not. In multiple South Slavic populations, including Serbs, mitochondrial DNA shows strong continuity with much older European lineages. Lineages that were already present in the Balkans long before early medieval migrations. Lineages tied to Neolithic and even pre-Neolithic populations. At first glance, this feels like a contradiction. Two genetic systems describing the same population and telling different stories. But it is not a contradiction. It is a record of process. Mitochondrial DNA moves through history differently. It passes from mother to child. It spreads more diffusely. It is less affected by small numbers of incoming males and more sensitive to long-term residents. When incoming male groups integrate into existing communities, their Y chromosomes can spread rapidly. Maternal lineages, by contrast, often reflect the deeper population base. This creates a specific genetic signature. Paternal uniformity paired with maternal diversity. Not replacement, absorption. Seen this way, the Serbian genetic profile stops looking puzzling. It starts looking historically realistic. Male-mediated migration does not require population disappearance. It requires contact, integration, and time. The genetics reflect that quietly, without drama. And this is where the emotional weight of the story shifts. Because many people instinctively treat origin as singular. One homeland, one movement, one founding moment. The maternal data refuses that simplicity. It shows that while new lineages arrived, older ones remained. That families formed across cultural boundaries. That identity emerged from interaction, not isolation. To make this more intuitive, think about language. Languages spread the way paternal lineages often do. They can be adopted rapidly. They can replace earlier speech without erasing the people who spoke it. DNA behaves similarly, but with more memory. A population can change its language, its political structure, even its name, while still carrying deep biological continuity through maternal lines. This does not weaken identity. It explains how identities survive change. There's another detail here that matters, and it often gets misunderstood. Maternal diversity does not mean randomness. It shows structure, regional patterns, signals consistent with long-term settlement in the Balkans. This is why geneticists talk about stratification rather than mixture as chaos. Different layers reflect different time depths. Recent movements sit on top of older foundations. In some cases, this layering has consequences that science still studies today. Small, relatively isolated communities can carry rare genetic traits at higher frequencies. This is not a flaw or a stigma. It is a known effect of population history, often called the founder effect. One or two examples appear in the Balkans, as they do everywhere else in the world. They matter not for identity, But for medicine, they help researchers understand inheritance, risk, and prevention. Here again, genetics is descriptive, not judgmental. What matters for this story is the logic it reveals. If the paternal line reflects movement and the maternal line reflects continuity, then modern populations are not the result of a single event. They are the result of interaction between arrivals and residents. This reframes the idea of shocking origin. The shock is not that Serbs carry multiple ancestries. That is normal. The shock is how clearly different genetic systems preserve different chapters of the same history, and how often those chapters are confused or collapsed into one. Once this distinction is understood, many false debates dissolve. There is no need to argue between migration and continuity. Both are present. Both are measurable. And once that becomes clear, a new question opens. If this layered pattern appears in a region as interconnected as the Balkans, what happens in populations that developed under very different conditions? Communities shaped by isolation, geography, or strict social boundaries. Because if mixing explains one story, then its absence, or its limits, should explain another. That is where the investigation goes next. Once you understand the logic of layered ancestry, the Serbian case stops looking unique. It becomes a reference point, because the same genetic principles appear again and again, but they behave differently depending on geography, history, and social structure. Look at communities shaped by isolation rather than crossroads. In parts of Ethiopia, long-term continuity dominates genetic patterns. Migration exists, but its impact is uneven. Local lineages remain strong because geography limited large-scale replacement. In parts of India, the signal shifts again. Here, ancestry layers reflect thousands of years of structure, not just movement, but endogamy, caste boundaries, and regional separation. Genetics does not contradict identity. It mirrors how identity was maintained. In East Asia, including China, yet another pattern appears. Large populations, deep continuity, and expansion that absorb newcomers rather than being reshaped by them. The balance between paternal and maternal signals looks different, not because the science changes, but because history does. These comparisons matter because they remove the illusion of exception. Serbs are not an anomaly. They are a clear example of how populations form in contact zones. This brings us to the core synthesis. Genetic ancestry is not a single thread. It is a mosaic. Some pieces reflect movement. Some reflect staying. Some reflect neighbors becoming family over time. The mistake is not in telling origin stories. The mistake is in telling them as if they had only one dimension. Here is the sentence that holds the entire video together. Identity is not written in one genetic line. It emerges where DNA, history, and memory intersect. The paternal line shows how influence arrives. The maternal line shows what endures. Culture explains how those elements fuse into something new. This is why genetics cannot replace history, and why history cannot ignore genetics. Each without the other produces a flatter version of reality. Now we can close the loops we opened at the beginning. Yes, one genetic line points clearly toward Slavic expansion. And yes, another preserves deep Balkan continuity. Both are real. Neither cancels the other. The origin does not split into competing truths. It splits into complementary ones. And this is the final shift in perspective. The question, who are we, cannot be answered by DNA alone. But DNA makes it impossible to pretend the answer was ever simple. History is written in books. Biology is written in cells. Identity lives in the space between them. If you want to explore more stories where genetics quietly dismantles easy explanations of origin, subscribe.